Hi, welcome to Hard-Boiled Synthesis, Lecture 14. We're back. It's been a while. I had a nice break. I feel energized, and I got a surprise for you guys. I finished the extractions, and so today's lecture, I'm just going to talk about what came out of it. Diversity of outcomes. It's not going to be an incredibly long lecture, but it's the start to actually getting the project completed. I think that's the most difficult stage, is coding and extracting. Not quite done yet. I still have to compute the effect sizes, but I'm ready to talk about what's been extracted, diversity of outcomes, and how we proceed from this point forward. Now, I'd just like to first off say that it wasn't easy to extract outcomes from all these studies. And I think that's just like a thing associated with repellency studies, repellency publications. They're very inconsistent. Even though we have the WHO and EPA standards and all sorts of things that researchers could use to standardize the protocol on how to test these things, everyone just kind of does variations of those things. And very few of the studies that I extracted data, laser in, are focused on testing catnip. Most of them, catnip is an ancillary thing that they want to test. They're either trying a new repellency strategy to, say, for example, infuse nets with different types of repellents, or they're interested in how other compounds can help disseminate the volatiles of a repellent, and they just use catnip. But let's just jump into it. I mean, what else is there for me to discuss? If you're new to the channel, my name is Marc Lajeunesse, and this has been a very long road to get to this point. 5 hours maybe of videos watching me extract data. I got an email from a colleague saying, "What are you doing? Nobody wants to watch that much time of someone just sitting down talking to himself extracting papers." And I agree. But I did it as an example of what is the most difficult and challenging part of the meta-analysis. Time-consuming part is sitting down, extracting data. And I'm still not quite done that yet, because I have yet to convert all those outcomes into proper effect sizes. You know, what I talked about in the last lecture, the data, the ingredients to a proper meta-analysis. Okay, well, I'm just glad that that phase is done. I certainly have a bunch of things that I'm going to add to my wall of deviations, walls of what I call my wall of shame, my deviations from best practices. I did not extract all the outcomes I could have. A lot of studies kind of have like this factorial, a multi-dimensional approach to testing repellents. Again, because catnip is ancillary to what they want. They want to devise like an optimal repellency application, and which means they're testing many layers of things in which catnip is part of that. Now, typically for me, I like to extract all that stuff. Like, even though, say for example, a treatment may include catnip and some other compound, I would normally still extract that, just because it does contain some information about catnip efficacy. But I didn't do that. I just lasered in and tried to identify all the contrasts, which is catnip by itself versus DEET or some other control thing. And I'm going to talk about the diversity of things. There's like not one study was consistent. And so what happens is we're going to have like a huge diversity of potential moderators, things that contribute to heterogeneity in experimental outcomes that I'll need to compare to see whether or not they impact negatively the way we could detect the efficacy of catnip as a repellent. And so let's just jump back to here. I'm like 95% done the extraction coding phase. Now it's just making sense of the outcomes and getting ready to actually compute the effect sizes. That's going to be the next lecture. Now I'm just going to talk about the diversity of outcomes. And to follow up on some of my conspiracy, hard-boiled stuff about the origin of 10 times, catnip is 10 times more effective than DEET, I figured out some more information. So if you remember, there was a patent associated with catnip, which seems to be the origin of this claim. Not the patent, but results reported in the patent were presented at a conference almost 20 years ago. That conference outcome got some heat from the press, got some attention. They did some reports, and that got disseminated in the social media, into the media, of this catnip is 10 times more effective than DEET. But the actual outcomes were locked into a thesis dissertation. And then going through my extractions, I found that they eventually did publish the thesis dissertation results more than 10 years after the original thesis. Apparently, what it seems like, after the patent expired, they published the results. And I got a little bit of criticisms associated with that study. It's not fully reported. Why jump ahead? I'm going to talk about it later. Right now, my goal is just to talk about diversity of stuff. Okay, so I was able to pull out numerical data from 21 studies that use catnip in one way as a repellent to mosquitoes. Out of those 21, 264 potential effect sizes can be calculated from these outcomes. I've yet to do that stage, so my gut feeling right now is this is like an upper level. 264 is the my fantasy data set, but the reality is once I start computing effect sizes from these outcomes, a lot of these are going to get dropped out, just because one thing that was super common was no variability in the outcomes. And that has to do a lot with how repellency is measured in these studies. When you're measuring things in terms of like percentages, there's an upper and lower bound. And if every replication yields 100% efficacy, there's no variability with that. So you could take an average of 10 100%, you're not going to get any variability that could be estimated from that. And so that makes it super tough to compute effect sizes, because you got zero variances, and that's not useful at all. It just makes for some awkward effects downstream that aren't really worthwhile to pursue, even though they do contain data, information useful for the meta-analysis. The barrier here is the way those data were reported. And so, for example, I did not extract numerical data from about 80 potential outcomes, because they were reported in a way that was not useful to compute an effect size. They were either reported already as an effect, and again, the goal of meta-analysis is to use a collection of effects that could work together as a common currency. But when studies report their own effects, their own derived effects, there's no way to convert it to what we could use for meta-analysis. A really common one that was reported was repellent efficacy. which is like the ratio standardized by the controls of the treatment. It's a mishmash of stuff that you can't recover any solid outcomes from. And I tried to see if I could do the algebra to rearrange things and estimate what the raw numerical data were from that, the counts, the means. It's just not possible. And those are just lost outcomes. And I feel like a deep criticism I have with these repellency studies is that they use these abstractions of repellency when really just showing the raw data is the most useful thing to make decisions. Anyway, okay, so potentially my data set will be around 260 outcomes to combine and compare. That's a lot of data. But there's huge variability in those outcomes, right? Not one of these 21 studies are the same. So for example, we have 15 different mosquito species tested, and some studies reported communities of mosquitoes, right? So they just, people are sitting outside and counting contact rates of mosquitoes, but they don't know the identity of these mosquitoes. Maybe they do, but on the spot like that, you're just counting landing times, probing times. If they go a step further, they may actually catch the mosquito and then try to identify it later. But these often get lumped as like many Anopheles species. To me, that's the ideal repellency test, is outside with a human, with a huge diversity of mosquitoes, right? Fed mosquitoes, starving mosquitoes, old mosquitoes, young mosquitoes, different species of mosquitoes, mosquitoes that weren't, that did not develop under ideal circumstances, right? They grew up in a crappy pond, right? So the adults may not be as amazing as a lab-reared mosquito that has unlimited ad libitum food its whole life, right? So you're getting a diversity of mosquitoes being exposed to repellent. But I imagine the reason there's a lot of like these standardized protocols with repellency tests is they're trying to remove some of that noise, try to make experiments consistent. But I think the more consistency is very valuable, right? Always using mosquitoes of a certain age, making sure that the mosquito is starved before you're doing your repellency test. These things are not consistently reported, unfortunately, but these would be ideal experimental designs. But what you're losing out is the mosquito really reflective of what you observe in the field? And observed in the field, it's messy. You're getting a collection of mosquitoes with different attractions, and so, but we do include some field studies, so I'm happy that there's those guys. It's not strictly specific tests of individual mosquito species, of like 5-day-old mosquito species. That's not really much of a criticism. I love seeing a diversity of species and mosquitoes. That's what we want, right? A good repellent acts great on a lot of different species. That's why DEET is awesome. It works great on many species. But my impression is, what I've seen from my extractions is that catnip is not consistently effective across many, many different species. Okay. In terms of like the trials, the designs in which repellency tests are conducted, man, this is a real diverse. This is where the real heterogeneity occurs. even though a lot of studies do the arm and cage trials, right? You have like catnip, DEET, sticking your arms in cages, looking at landing times. There's a whole bunch of other types of studies, which again, because are applied because testing catnip as a repellent straight up is not the goal of the study. Goal of the study is to distinguish between, say, for example, different volatile chemicals from catnip, right? You're teasing apart different chemicals, you're releasing them in the air. That's what you're interested in. And so this just means that there's a lot of potential sources of variability when comparing the outcomes of the how well catnip performs, because a lot of these studies are just kind of using a totally different approach to testing the repellent effects. And in fact, an outcome of this diversity of experimental designs is you get a huge diversity of responses measured. People are collecting different kinds of data. They may not be collecting probing time or landing time. They may be collecting things like that I excluded was like sensory activations, or whether or not the mosquitoes respond to different concentrations of volatiles in the air. And what gets reported is the concentration of the volatile that elicits the most response, and not the actual number of landings of a mosquito, right? Mosquitoes respond the best in terms of repellency at 3% catnip oil, rather than saying 80% of mosquitoes were repelled by the catnip oil at 8% concentration. And so this is where I'm going to get like a significant hang up in estimating the effect sizes, is because of this diversity of outcomes. Luckily, a lot of them are just counts, and so I can convert that into odds ratios. But other ones are means, averages, derived averages, unfortunately. So like, again, they're using their own effect sizes. But I think I got a huge diversity of stuff that I could aggregate. And so I'm not worried about it. It's just I'm nervous about approaching this stage, because it is kind of a boring one where you have to look at each individual effect and compute an effect size. This isn't like a medical research where the designs are standardized, right? So you could just jump into computing effects because you have your treatment control counts. No. This is all, every study, you have to spend some time to calculate effect sizes. I think I talked about everything I needed. There's also a huge diversity in concentrations of catnip, from like really small concentrations to some that are comparable to what the how do you say this? The WHO or the EPA suggested concentration for DEET in practice, which I think is 20%. And so you get catnip concentrations from like 20% all the way down to like fractions of a percent. And some of the funny results, right, to go back to this stuff right here, the origin of catnip being 10 times more effective than DEET, appear to be derived from experiments where the catnip and the DEET concentrations are super low. I mean, they're not reporting results at the 20% concentration mark. They're reporting results at the 0.11% mark. And so it kind of makes it difficult to understand like the strength of the claim that catnip is better than DEET when the concentrations are just so low to begin with. I'm going to talk a lot more about that study because I feel like a lot of it was just me trying to find the outcomes, and then they did publish the outcomes, and so I was able to kind of digest and think more about the quality of the test. And there's a lot of iffy stuff about it. I'm not criticizing the research per se, it's just the design of it seems not providing strong evidence for the claim. That's the whole point of the meta-analysis, though, is to include that study, include many other studies, and try to reach a consensus. What else did I want to talk about? So I talked about concentration, diversity. Oh, catnip. So there's huge diversity in how catnip itself is treated as a repellent. No plants, no like, here's a bush of catnip. Does it repel? None of that. Most of the studies are essential oils diluted to various concentrations. An additional layer on top of that is many studies extract the active compounds from the essential oil, the naphthalactones, which is an isomer. It comes in two variations. And so most studies extract the two of them and test those separately. Now there's an additional layer to those, which I had to make an executive decision, which might shoot myself in the foot at some point. But I decided to exclude these studies, were the studies that take the two isomers and hydrogenize them, right? So they further process those compounds into something called dihydronaphthalactone or something like that. Going back to this with the patents. If you remember, there was 2 patents originally filed for the essential oils and the naphthalactone, the 2 isomers, which have expired since, I don't remember, 2011, 2012. But there's an existing patent right now that's still in effect, and it's not the essential oil or the 2 isomers, the 2 active compounds, but the hydrogenated versions of those compounds. And it's held by DuPont. So they have some sort of plans of it using this, what I call, a derived chemical from the active compounds, the natural active compounds and essential oils. That's why I kind of decided to drop those studies, the studies that look at the hydrogenated version of the compounds. It seems too far removed from catnip, right? Is it still catnip at this point if you have manipulated the active compounds? Maybe that's some weird philosophical thing. Maybe a chemist could argue, well, you know what, it's just derived, you should include it. I decided to drop it just because it seems like multiple steps away from just catnip. Not really multiple steps. You have catnip, essential oil, right? It's already been processed. Compounds focused into an oil. Then you have your separation of the compounds, right, using some HPLC thing. Then you got your 2 isomers, and then those get treated with hydrogen into these now patented compounds used as repellents. And there's been a bunch of those. I didn't quite look at the institutions involved in those projects, but I think quickly eyeball it. It was always related to DuPont, which makes sense because they have the patent to it. So I dropped that stuff. I imagine I'm going to have to leave some discussion on that when I pull together the manuscript on why I decided to exclude these things. I just felt like it was just too much. The study's already becoming too much for me to report, for me to describe. All I want to do is answer that claim. that whether or not catnip is 10 times more effective than DEET. And now I'm getting into all sorts of like these derived compounds. It's just, maybe someone could do their thesis on this, but I'm not ready to take it that far. I just want to keep it fairly straightforward to get this project done. And so, timeline. Okay. Timeline. I talked a lot already. Timeline. Okay, so I don't envision many stages left. There's the stage in which I have to compute the effect sizes from the extracted outcomes. That should be one lecture. Once the effect sizes are computed, then we can start actually modeling the effects using meta-analysis. That might be 1 or 2 lectures. Again, what I want to test is very narrow. I'm not going to do a full exploration of heterogeneity in effects. I'm going to hit on these key parts so that I can roll it into the final stage, which is pulling together the manuscript. This is kind of what happens with these meta-analyses, is there's a lot of opportunities to blow up the project, right? As you extract data, you see relationships that could be tested, dimensions that could be evaluated in terms of methodological heterogeneity, or just like heterogeneity that is associated with the biology of the plants, like the plant chemistry stuff, or heterogeneity associated with the mosquitoes, right? Different species of mosquitoes. I mean, how the mosquitoes perceive the repellent. I had to exclude a bunch of those studies. Some of them were pretty cool. I mean, there was like a mutilation study where they just kind of removed the palps and the antenna of the mosquitoes and tested whether or not it was still repelled, trying to figure out which organ on the face of the mosquito is sensing the repellent. And the bummer of that study, just as an aside, was they didn't include an unmutilated mosquito as a reference baseline comparison. I just thought it was so odd, right? There's so many organs to remove, right? You got your multiple mouth things, you got your antenna, but then there's no like unmutilated mosquito to compare to with. Anyway. Okay, so next step for me, computing effects, which is going to be slightly ugly, but I see the end of the tunnel here, which I did not have a clear vision of last week when I was still extracting data. I was like, oh no, this is going to take forever. But I'm done that worst phase, and so now we should be able to zip through fairly quickly. So I'm going to end it with that today. Thanks for watching. And I'll see you guys tomorrow.